Histamine, derived from the Greek word histos, meaning tissue, is a naturally occurring compound found in animal tissues and certain plants like stinging nettle. Its pharmacological properties were extensively studied by Dale in the early 20th century, revealing its significant role in allergic reactions and tissue injury responses. Today, histamine is recognized for its crucial physiological functions. Histamine is predominantly stored in the granules of mast cells, with high concentrations in tissues such as the skin, gastric and intestinal mucosa, lungs, liver, and placenta. Non-mast cell histamine is found in the brain, epidermis, gastric mucosa, and growing regions. The turnover rate of histamine varies, being slow in mast cells and fast in non-mast cells. Additionally, histamine is present in blood, body secretions, venoms, and pathological fluids. Histamine, chemically known as B-imidazoline-lethylamine, is synthesized from the amino acid histidine and rapidly degraded by oxidation and methylation. In mast cells, histamine is stored within intracellular granules bound by an acidic protein and heparin. Upon exocytosis, sodium ions in the extracellular fluid exchange with histamine, releasing it. Increased intracellular cyclic adenosine monophosphate, or CAMP, levels, caused by beta-adrenergic agonists and methylxanthines, inhibit histamine release. Orally ingested histamine is inactive, as it is degraded by the liver before absorption from the intestines. Histamine exerts its effects through 4 types of histaminergic receptors: H1, H2, H3, and H4. These receptors were classified by Ash and Schild in 1966, with H1 receptors being blocked by then-available antihistamines. Sir James Black developed the first H2 blocker, burimamide, in 1972, confirming this classification. The H3 receptor, identified in 1983, primarily functions as an autoreceptor controlling histamine release in the brain. The H4 receptor, discovered in 2001, shares considerable homology with the H3 receptor and binds many H3 ligands. H1 receptors are primarily involved in allergic reactions and are targeted by common antihistamines. H2 receptors, on the other hand, are found in the stomach lining and regulate gastric acid secretion. The development of H2 blockers such as burimamide revolutionized the treatment of peptic ulcers and other acid-related disorders. H3 receptors serve as autoreceptors in the brain, modulating the release of histamine and other neurotransmitters. Although selective H3 agonists and antagonists have been developed, they have not yet found clinical application. The H4 receptor, expressed mainly in eosinophils, mast cells, and basophils, plays a role in allergic inflammation. Activation of H4 receptors enhances chemotaxis of these cells, and H4 antagonists are being explored as potential treatments for allergic inflammatory conditions like rhinitis and asthma. Understanding histamine and its receptors has profound clinical implications. From treating allergies with antihistamines to managing gastric acid-related conditions with H2 blockers, histamine research continues to influence modern medicine. The ongoing exploration of H4 antagonists holds promise for new therapeutic approaches in allergic inflammation.